Hello, this is Dr. Barker. Welcome to the fall 2022 version of my immunology course. This is a version of Lecture 1. However, the actual audio of Lecture 1 did not work. So I ended up needing to re-record the Lecture 1 audio at a later date. This is an approximation of what I said during Lecture 1. I apologize if it doesn't match your notes. At the beginning of this lecture, I first spent some time discussing course policies and the syllabus, which the students in my class will of course have access to. We then got into small groups so that the students could brainstorm some aspects of a definition of immunology, as well as thinking about ways that their immune systems might influence their lives. And we then had a good discussion of those topics. I pointed out that we can see some observations of immunology that historically are present for a very long time. One such example is this quote from Thucydides, though there are also examples in ancient Chinese and ancient Sanskrit literature. Here you can see that Thucydides is quoted as noting, though many lay unburied, birds and beasts would not touch them or died after tasting them. The bodies of dying men lay one upon another. But those who had recovered from the disease had now no fear for themselves, for the same man was never attacked twice, at least not fatally. This is a quote from the 400s BC, and as you might guess, Thucydides does not have any of the prerequisite material that I require for this course. However, you can also see that he was able to make an important immunological observation. This observation that the second time one was exposed to a particular microbe, there was a different outcome than the first exposure. This is one sort of interesting part of immunology in that because we all have such experience with our immune systems and immunological phenomena, we are able to make some observations. So no matter your amount of biology background, you are able to make some amount of immunologic observation, just as you see here with Thucydides. We can also see this when we think about the experiment that is generally cited as the first ever immunologic experiment. This experiment was done by Edward Jenner in 1796 and published in 1798. The story goes, we now know that some parts of this story are a bit not true, but the story goes that Jenner saw the skin of some lovely milkmaids who had not been infected with smallpox, or at least affected by smallpox, and he surmised that perhaps infection with cowpox led to protection against smallpox. He then went ahead and did an experiment with an 8-year-old boy, James Phipps, as you can see here, where he first infected James Phipps with cowpox and then infected him with smallpox. Yes, that is an incredibly unethical experiment that we would never be able to do today, but he did show that James Phipps was protected. This sort of experiment led to pretty dramatic outcomes. We were able to see this infectious disease that had killed about a third of those infected, at least 300 million deaths in the 20th century, and many famous and individuals or those who had historical importance all be we were able to see this based on this first immunological experiment where there was no knowledge of general biological prerequisite material eradicate this disease to the point where this virus was listed by the World Health Organization as having been eradicated from Earth on December 9th, 1979. So this very simple first immunologic experiment has important ramifications and important outcomes to this day. So based on this, what we can see is that people are able to make immunological observations for a long period of time. And in one sense, we all have some level of understanding of the immune system. However, if we also think about some other aspects of the immune system, there's another thing to think about. When other biomedical scientists think about immunology, they say things like immunology is where intuition goes to die. Because immunology has some aspects that can be a bit counterintuitive, and that can be sort of unexpected and can challenge your basic understanding of some aspects of biology. In reality, immunology doesn't violate anything, but it does really require you to understand the basics of biology and what's really going on behind them. Well, it will uncover many of your misconceptions about the immune system. If we look at this article, which is one of my favorite articles about immunology, there's a part that mentions this joke. There's a joke about immunology, with Jessica Metcalf of Princeton recently told me. An immunologist and a cardiologist are kidnapped. The kidnappers threaten to shoot one of them, but promise to spare whoever has made the greater contribution to humanity. The cardiologist says, well, I've identified drugs that have saved the lives of millions of people. Impressed, the kidnappers turn to the immunologist. What have you done, they ask. The immunologist says, the thing is, the immune system is very complicated. And the cardiologist says, just shoot me now. And again, this is both true in that in immunology, the complication starts to come in the details. And we need to pay attention to those details. And it can be quite complex and not just the simple thing that everyone has observed from their daily lives. This is also, perhaps regrettably, the view that some of the other biomedical sciences have of immunology, where we always come up with this idea of, oh, it's so complicated. So be aware of this and be ready for this semester as we study this phenomenon, and realize that we're going to really need to pay close attention to many of the details. The author of this article, Ed Yong, who's one of my favorite science writers, also wrote in a tweet, if there was an immunology board game, it would have a rule book that's 600 pages long and be written only in acronyms. There would be a cupboard's worth of tokens, and every so often the board would spontaneously catch fire. So just be aware that this is one of the aspects of immunology you're going to have to be ready for. There are lots of great reasons to study immunology. One, it's very vital to health and protects you against pathogens. Malfunctions can lead to serious illness. It's fascinating and complex. It has a long history and we know a lot, yet there's still more that we don't know. We're going to see a lot of important biological principles. We're going to see techniques that are important in other areas of biology. We're going to see a system that's full of interconnected parts and that will integrate other parts of biology. But you also have to realize this is a fascinating subject, and every day we are going to come upon some question where we don't know the answer. You will ask questions, and I will say that's a thing that researchers are trying to look at. We are going to be integrating a lot of other biology courses. I know what the prerequisites for this course are. I don't expect that you have taken other biology beyond that, but we are going to be pulling in lots of biology from other places. We are going to be looking at a lot of different interconnected parts. As you will see, immunologists love our nomenclature. We love our acronyms. We're going to have to pay close attention to the nomenclature and acronyms. And sometimes things are a bit counterintuitive. As I said before, we are never going to be violating any major biological principles, but we are going to potentially expose some misconceptions in those basic biological principles that will make things a little bit difficult if you're not able to be flexible in your thinking. So these are some of the reasons why studying immunology can be a bit challenging. But there are also some reasons why teaching immunology can be a bit challenging. So this is an example of how many of us think about the immune system. And you can see this nice, big, interconnected network of many different cells interacting with one another. Somehow, what I have to do in teaching this semester, in putting together a syllabus, in trying to put together all of the things we'll be talking about, is take this interconnected network and flatten it out and make it into a linear syllabus. What you can see is I can't really come up with a way to pick one piece of this immune response and teach it to you without invoking or mentioning some other parts, because all of the other parts are connected. There's no place where you can really start without having to think about connections to some other pieces. So in lecture 2, we're going to be talking about cells and organs of the immune system, and we can think about it as sort of the cast of characters of the immune system, where you can start to see all of the different players and names, so that as I start to tell you about some of these different pieces, I can at least tell you which other pieces they are linked to. When we think about the immune system, your textbook defines the immune system or immunology as the study of the physiological mechanisms that humans and other mammals use to defend their bodies from invasion by other organisms. That is absolutely true. For the most part this semester, we are going to be talking about the vertebrate immune system, and we will really be focusing on responses that are made by humans and other mammals. We will have a few situations where I will talk about how we defend against invasion in non-mammalian organisms, though immunologists, particularly in a course like this, tend to think about mammals. And we can imagine lots of reasons why it's really important to defend ourselves from invasion with other organisms. Those other organisms, of course, are going to really want to use a lot of our biomolecules and other parts of our body as nutrients, as a nice, safe, temperature-controlled, good humidity, good other types of chemical environments home. And so we can certainly imagine some reasons why it might be important to defend ourselves from these other organisms, why it is advantageous for them to want to use parts of our body. And so we can also think a little bit about some of the issues with this defense. So yes, the pathogen is potentially going to cause some damage to the host. And if we think about why we should defend ourselves, that makes sense that we don't want to allow damage to happen. But we can also think about reasons why defense might be a bad thing. So sure, it's a great idea to defend ourselves against a pathogen. We defend ourselves using a number of different host resistance mechanisms. But what we should also remember is that those host resistance mechanisms also can have a little bit of a downside. Those host resistance mechanisms can also potentially damage the host. So we need to make sure that we think about different types of host defense mechanisms, and remember that there is this con of some additional damage that may be caused by our defense mechanisms. And we need to make sure that we walk the line of having defense that damages pathogens without causing too much damage to the host. So that already tells us one place where we're going to need to think about regulation. Immunologists love to think about Goldilocks. We don't want to have too little of a response because we'll have damage from the pathogen, but we don't want to have too much of a response because we might have damage from the immune system. That response needs to be just right. If we look in addition at some other pros and cons, realize that when we are making an immune response, which is listed here as presence of insult, the majority of our resources, shown in the darker pink here, end up going towards defense. The cell stops trying to grow, or the organism stops trying to grow. The organism stops trying to reproduce. The organism basically goes towards solely spending its ATPs on defense. And while that's great in terms of saving oneself from a microbe, it's kind of problematic in all of the other biological functions that the organism might want to do. Defense is extremely expensive biologically. And so, again, we need to have a bit of a trade-off. We don't want to spend all of our resources on defense at the expense of other types of biological functions, but we also don't want to have too much damage by the pathogen. And so as a result, the immune response has very much evolved to balance the damage by the pathogen and how much damage we can take versus the amount of host resistance. Sometimes we might actually have a less than ideal response to the pathogen in terms of getting rid of the pathogen, but that will have the perk of allowing us to avoid damage or giving us the ability to save some of those metabolic resources that might have otherwise been used for other purposes. This is related to an incredibly important set of phenomena that we need to talk about and that we'll see over and over again this semester, which is the difference between a state known as infection and a state of disease. Infection is defined as a situation in which a microorganism is established and growing in a host. So if there is a microbe in your body that is starting to reproduce, that is infection. It has nothing to do with whether or not you're seeing any damage to the host, whether we have any type of physiologic changes. Alternatively, disease is when we have some kind of damage or injury to the host that impairs host function. And when that disease is related to some kind of microorganism, some kind of pathogen, then we will refer to that as infectious disease. So realize that the key thing here is that we want to avoid disease. There are many situations where you are infected with a microorganism where you are not harmed at all. Your GI tract right now is full of a number of different bacteria that are actually helping you with metabolism, with making some vitamins. They're helping with some immune processes, a number of different things. But you are infected with all of them. They are growing and established inside of your body. However, they don't cause disease. In fact, they're doing something positive. So what you want to realize here is that infection does not always lead to disease. Similarly, just because you're exposed to a microbe, that doesn't mean you're necessarily going to be infected with that microbe. I point this out because we should realize that sometimes the goal of immunity is not to completely protect us from infection. Perhaps protecting us from infection would be just too high of a bar. That might put us in danger of having an immune response that might cause damage to the host, or an immune response that might be metabolically just too much. But if instead we simply try to have an immune response that protects us from disease, that might be a little bit of a lower bar, and that might be a better answer to some of these trade-offs. So our immune response is not always solely about protecting us against infection. It may, in some cases, be a situation where we're protecting against disease. At the bottom, I'm showing you a image that shows some of the differences between the events that need to happen from exposure to the point of actually getting to disease. And what I want you to realize is that there are multiple different events that need to occur between when you are first exposed to a pathogen and when that pathogen leads to disease. So it's not some automatic link between when you are first exposed to a microbe and you having some kind of disease. You need to get infection, and then there needs to be additional events that will happen in order to lead to disease. When we actually think about defending ourselves against different microbes, there are a number of different events and different things that we can think about in terms of how we can imagine those targets. We can imagine what might be different, for example, between our cells and microbial cells, or between our cells and other types of non-cellular microbial pathogens. And so just like if you were trying to come up with a surveillance system like the TSA has, you might think about, well, what types of things might be indicative of a problem. We can think about similar types of things of what types of issues are we going to imagine as being indicative of a microbe. We need to think about a few different properties of microbes in terms of thinking about aspects of the immune system and immune defense that we might need. One of the things that we will need to think a little bit about is growth rate. So here you can see a plot that is very common in my microbiology class. Those of you who've taken my microbiology class will recognize this plot, where we're looking at a plot of bacterial growth rate. We have the traditional bacterial growth phases over time. And so you can see the number of bacteria on the y-axis and time on the x-axis, and you can see how quickly those bacteria are doubling. If you recall from microbiology, we think about the standard growth time, or the generation time of E. coli, for example, as about 20 minutes. So we go from 1 cell to 2 cells in about 20 minutes. You can see that here in about 24 hours, we would go from 10 to the 5th, or 100,000 cells, up to 10 to the 9th, or about 1 billion cells, looking at a typical growth rate of bacterial cells. Now we can compare this to thinking about the growth rate of eukaryotic cells like your cells. Your cells are able to undergo 1 cell division or 1 cell cycle at the fastest in about 24 hours. So you're able to go from 1 cell to 2 cells in 24 hours. E. coli, as I said, will go from 1 cell to 2 cell in about 20 minutes. And you can see that in 24 hours, the E. coli would go from 10 to the 5th to 10 to the 9th cells. Therefore, if we were not able to do anything about the numbers of these cells, E. coli would always win, and microbes would always win, because they're always able to outreplicate us. And so sometimes we have some immune defenses that are simply defenses that will stop replication, to slow down replication of different microbes and to give us a chance against them. This difference in growth rate is even more dramatic if we think about, say, viruses, where in the case of a viral life cycle, we might go from 1 viral particle to thousands or millions of viral particles, all within 1 generation. So this is one thing that we need to think about when we're trying to imagine what our immune system must defend against. Simply, this numbers problem is one. When we look at different microbes, there are also huge differences in the size of those microbes. You can see viruses and different types of bacteria here with their size, as well as fungi and other types of parasites. You can imagine that something that might combat a virus at 10 to the negative 7th meters might be different than something that might combat a parasite at 10 to the minus 2 meters. You could particularly imagine that with regard to something like phagocytosis, whether one of our cells will be able to engulf or eat one of these microbes. The size of the microbe relative to our cell is going to make a big difference. At the bottom, you can also see an example of another pathogen, Ascaris. This is a parasitic worm, and you can see this is Ascaris that has come from someone's GI tract, and it is being held in someone's hands. You can see the hands here, and you know the size of hands so that you can kind of get an idea of the size of the worms here. You will probably realize quite quickly that we would not be able to phagocytose those worms. We can also think about things like whether or not some of these microbes provide unique targets to our cells or are similar to our cells. For example, fungi and different types of parasites are eukaryotes, and so their cells are more similar to ours than are the prokaryotic cells like bacteria and archaea, which thus would provide some more unique targets. Viruses will use many of our cellular enzymes and other cellular processes, which makes it somewhat difficult to target those organisms without targeting our cells. We can also think about things like where these organisms are. Are they outside of our cells, inside of our cells, in different compartments? All of those things will influence the types of defense that we'll be able to make. We'll see some additional parts of this as we go throughout this semester, and this will be the end of lecture 1. I apologize for the awkwardness of this lecture when I was recording it after the fact.